Thanks everyone for coming. We're going to get going around 9:05.
Which is 12:05 for us here in Montreal.
Yes, sorry on the West Coast here. Welcome everyone. I'm Signe, a PhD student at the University of British Columbia and a member of the CEEHRC trainee committee. This is session 1 of our trainee-led seminar series on epigenomic profiling, a deep dive into chromatin mapping technologies. Today, Wajih will be giving a presentation on the experimental design and execution side of CUT&RUN and CUT&TAG technologies. Wajih is a PhD candidate at McGill University and a research trainee in the labs of Drs. Nada Jabato and Livia Garcia at the RIMUHC. Wajih's PhD work focuses on epigenomic dysregulation in pediatric sarcomas and gliomas to unravel disease mechanisms and targetable dependencies. A reminder, if you have any questions during or following the talk, please enter them in the Q&A section on Teams. Wajih will answer questions at the end of the presentation. Take it away, Wajih.
Thank you, Signe. So hello and welcome everyone to the first session of our workshop series, and I'm happy to take you on our dive into epigenomic technologies. If you're new to epigenetics, this is for you. And if you're not new to epigenetics, this is also for you. We've got you covered. So the field of epigenetics taught us a lot about life in the context of health and disease. It taught us about the tail of the histone tail and how every amino acid matters. Whether this amino acid undergoes post-translational modifications or PTMs, such as lysine 27, or is not chemically modified, such as glycine 34. Because specific changes on specific residues often result in severe consequences that are also specific to the tissue of origin where it had occurred, many of which also occur within tight developmental windows, leading to cancers in children, adolescents, and young adults. But of course, epigenetics also regulates many developmental processes, even back to early embryogenesis, where we already know that DNA imprinting has an essential role for proper development. But also, we are more and more appreciating the other forms of imprinting that also occurs on chromatin under the action of different epigenetic complexes. And here, of course, PRC2 is just an example of these mechanisms. Hence, the epigenome is composed of multiple regulatory layers that we map and study, from direct modifications of DNA to chemical modifications of histones, both the histone tail and the histone core, and of course, long non-coding RNA. Collectively, this layer modifies or influences chromatin architecture and gene expression. So, if someone were to search or ask for a definition of epigenetics, they might find very different things. But it's essentially the way the genome is specifically tailored depending on our different state of development, health, disease, and beyond. And in this workshop, we will focus specifically on modalities that's going to interrogate histones, PTMs, and DNA-bound proteins, such as transcription factors, but also chromatin modifying complexes. And these modalities would allow us to study specific histone functions, for example, to elucidate disease mechanisms endowed by oncohistones, which are cancer-driving histone mutants, but also naturally occurring histone variants, and how they regulate gene expression, genome integrity, and cell physiology. Moreover, it allows us to understand these chemical modifications we often refer to as PTMs, and how they interact with the epigenetic repertoire of histone readers, writers, and erasers, and how they collectively influence gene expression. So a lot of our current understanding of epigenetics actually came from ChIP-seq, or chromatin immunoprecipitation, linked with next-generation sequencing. The protocol actually starts off with some form of cross-linking, but sometimes it's also performed under native conditions. Of course, with each variation has its own trade-off. Next, chromatin is isolated and sheared down to a few hundred base pairs, followed by the actual IP step. Specific antibodies are added to bind your target, followed by the addition of protein A or protein G coated magnetic beads. And then from this IP, the chromatin, of course, DNA can be purified and then can also be sent for sequencing to look at genome-wide deposition patterns, or to simply interrogate the epigenetic state of specific loci by some form of PCR. But of course, just like every technique, ChIP-seq comes with caveats. The problem actually starts with the very first steps of cross-linking, which notoriously produces artifacts. And that becomes even more problematic at highly transcribed loci. And then the issue continues when shearing chromatin, as this contributes to a lot of the noise coming from short sticky DNA fragments generated in this step. And then this forces us to perform a few stringent washes, which in turn also leads to some loss of signal. Collectively, this leads to a low signal-to-noise ratio, which ChIP-seq is infamous for. And that is one reason why this technique requires a high cell input, which actually becomes a limitation when you're studying rare cell populations, for example. So if you're aimed to profile specific immune cells, or let's say study satellite cells in response to tissue injury, or even when working with cells with limited proliferative capacity, such as stem cells coming from patients, or even stem cell lines that are undergoing the process of differentiation. And more critically, of course, it becomes a limitation when dealing with precious samples that cannot be replaced, such as patient samples. And of course, because of this low dynamic range, this technique also necessitates higher sequencing depth, which also means increased costs. Not to mention that this process can be very laborious, and a lot of sample-specific optimization is required, which can impose some restrictions, or at least some challenges as to the type and scale of the experiments that can be performed. Interestingly, because of the high noise and artifacts that ChIP-seq produces, ENCODE and others have also compiled a blacklist of loci that contain regions that exhibit high and unstructured signal, regardless of cell type and protein target. Newer modalities such as CUT&RUN, which stands for cleavage under target and release using nuclease, aim to address this issue. CUT&RUN can be simply considered as a more modernized version of CHIC or chromatin immunocleavage that allows for greater scalability and enhanced signal-to-noise ratio. And of course, it can be applied genome-wide. The technique was first introduced in 2017 by the Hennekoff lab, and many adaptations and modified versions were released since. In a nutshell, the live cells are first immobilized on lectin-coated beads, known as CUT&RUN beads, under native conditions. Then cells are permeabilized, most commonly with digitonin, and then incubated with a primary antibody overnight. Next day, protein A, protein G fused micrococcal nuclease, which is an exoendonuclease, is added. The MNase identifies the IgG portions of your antibody thanks to the protein A, protein G component. And then upon the addition of calcium, the reaction starts within seconds, cleaving your target-bound DNA, which can then be released to the supernatant. And then from these complexes, DNA can finally be purified, either with conventional organic methods or with spin filter column purification. And now I'll discuss the protocol in more depth and provide finer details and tips for experimental success. So as I said, in the first step, cells are collected in their native state, so they're living cells, and they are immobilized on these CUT&RUN beads. So in this step, the cell health is very important because cells are still alive. So activate your beads and prepare your buffers before cell collection, because you want to minimize the amount of time that your cells are not in their regular growth conditions. And of course, aim for the healthiest, most viable cells possible. So check viability upon collection. You can use a very simple trypan blue exclusion assay to make sure that the membrane is intact and the cells are live. And of course, be gentle. So be gentle with your pipetting, be gentle when you're handling them, because again, they're still live cells. And the cell membrane integrity is very important for this process, because this lectin binds the cells through glycated motifs on the cell membrane. So the cell membrane has to be intact during this process. Of course, cell numbers are also important. There are different modifications for the protocol. However, cell number is important using standardized protocols. Most commercial protocols and online protocols recommend 5,000 to 500,000 cells. Of course, if you have cells in excess, I would recommend that you stick to 500,000. And in case you need to do additional washes because you want to get rid of other contaminants from your cell culture media, because the cells are clumpy, because you did some form of cross-linking. So in case of these additional washes, account for losses that would occur. So prepare 20% more. And then the same way you want your cells to be as healthy as possible, make sure your beads are as functional as possible. So avoid any unnecessary drying, which might compromise their functionality. Keep them on ice and keep them in suspension as long as you can. And avoid overly incubating them on the magnet. Even if functionality is not greatly compromised or not even compromised in some cases, from experiments, we know that keeping them for too long on magnets causes them to overly aggregate and become harder to work with. So after your cells have been collected, you validated or you verified that they're healthy, and you bound them to the beads, so the beads absorb those cells, you can start adding your antibody. Antibody incubation, of course, occurs at low temperatures between 0 and 4 degrees. And in this case, the cell is permeabilized with digitonin because the antibody already has digitonin. And one of the important recommendations here is to maintain as much consistency as possible. So prepare an antibody buffer master mix before starting this process. So let's say in your strip tube, if the first 4, first 5, even first 2 samples are going to be used to map K27AC or K27 methylation, whatever the mark is, make one master mix that has the antibodies. And this way you make sure that everything is as consistent and as controlled as possible. Most protocols recommend 0.5 microgram of primary antibody, but in many cases, if your target of interest is lowly available or lowly abundant, you might need to add more antibody. So just follow manufacturer recommendations in most cases, and of course you can do your own optimization for the antibody concentration at this step. Incubation time is usually done overnight using standard protocol. However, some papers do show that incubating for a bit longer, maybe like 24 hours or more, might result in more consistent results, because I think the reaction would be more saturated in this case. And of course, keep the cells, keep everything on ice with caps closed. You don't want to have any accidents or any mixing, especially at this step, because your samples are still not indexed. And another note for incubation time and for the primary antibody, in most cases, using standardized protocols would work very well for most PTMs or for most targets, but you might have to optimize. And I'm going to provide some few resources at the end of the presentation. So next day, you're going to have your permeabilized cell, antibodies bound to your targets. So you're going to perform a few washes to get rid of any antibody that's not bound, followed by the addition of the protein AG fused MNAs. And then finally, calcium is added as a cofactor to activate the reaction. Upon the addition of calcium, the MNAs starts cleavage within seconds. And then your cleaved complexes carrying your DNA can be then released to the supernatant. So this step is also very easy. Actually, the whole protocol is very straightforward. It only requires a few washes, making sure that no mistakes are happening, and of course, maintaining consistency, which is something I'm going to stress on again here. So prepare your buffer master mixes with protein AG, MNases, and the other master mix with CaCl2 to make sure that all of the samples are treated equally. Again, all the washes here are performed with ice-cold buffers. You want your reaction at these steps to be low and slow, at low temperatures for a longer period of time, and this also prevents MNase diffusion on DNA, which would otherwise contribute to signal coming from background. And of course, again, stick to the recommended incubation times. Standard protocols work for most marks or for most targets, and you can optimize in case something doesn't work as well for your specific target. And then library prep can be performed after you purify your DNA from these complexes. In my own experimentation, I haven't seen any samples being compromised if I left them overnight. So you can leave your sample overnight in the fridge, but at least purify. So purify the DNA, and then you can incubate and do library prep next day. Cut and tag, or cleavage under targets and tagmentation, also offers a very similar workflow, but of course with different incubation times and different buffers. So I won't be very redundant here in explaining these steps. I would say the major difference here is the use of the TN5 transposases that are preloaded with sequencing adapters. And that allows for a direct to PCR approach without the need for purification or the early steps that we do during library prep. So in this protocol, nuclei are most commonly used to avoid sharing of mitochondrial DNA. You don't want to include those in your experiment. So the nuclei are bound to CONA beads. The primary antibody is again added overnight. And then next day, a secondary antibody is added. After secondary antibody addition, a few washes are performed with high stringency buffers that have higher salt concentrations to prevent any non-specific TN5 activity. Then the PAG fused the TN5 is added, which is going to bind the secondary antibody bound to your target. Then upon the addition of magnesium, TN5 will simultaneously cleave the target-bound DNA and add sequencing adapters in a process called tagmentation, which is actually very cool. And then from there, DNA can be released using STS and high temperature incubation. And then DNA can be indexed and sent for sequencing. So it's a direct to PCR approach. As you saw here, cut and tag essentially bypasses the library prep step. So these were the different experimental steps and a few tips for success. Most of it was making sure no mistakes happen, upholding consistency as much as possible. And of course, what's very pivotal, what's very important is to make sure that your cells are healthy, and same goes to your nuclei. For both cells and nuclei, you can use trypan blue exclusion assay or any other fluorescence-based method. So next, I'll discuss important considerations to get the most out of your experiments, because eventually data interpretation is our goal from doing these experiments. So one of the very important considerations, regardless of whether you're doing cut-and-run, cut-and-tag, or ChIP-seq, is spike-in normalization. So spike-in is a technique to calibrate. It's used to calibrate data across different samples by adding a known and constant amount of exogenous non-target material, which we often refer to as a spike-in. And this exogenous material has to be added to all of your samples, including your non-IP input. So the chromatin without the IP step or before IP. The spike-in is conventionally in a form of chromatin or cells or nuclei. In this cartoon, I used cartoon exogenous reference of cells. Of course, they have to be from a different species, so you can discern your spike-in from your target cells or your target genome more specifically. And the spike-in is very important, again, to calibrate. I'll show you a few examples to kind of simplify in case you haven't done spike-in normalization in the past, or you're still new to this technique, or new to epigenetics. So let's assume this example, you have two different populations of cells. You have the yellow cell population, which is untreated or treated with vehicle, and then you have the red population. So this population could be treated with an enzyme inhibitor, could be any histone writer or eraser. So it could be SETD2, EZH2, DOT1L, could be anything. It can also be cells that were infected with a virus or treated with an shRNA. Regardless of your setup, it's quite often that the cells will have a bit of a heterogeneous response, especially if you're working with a pool of cells. So in the absence of the spike-in, and because our protocols are all based on enrichment, we might not be able to detect actual changes. Again, because ChIP-seq and the immunocleavage-based assays all rely on an enrichment process. So let's say if these cells were treated with tazemetostat, an EZH2 inhibitor, and half of your cells lose K27 tri, CUT&RUN, CUT&TAG, and ChIP-seq are not going to enrich for K27 tri depletion. It's going to enrich for K27 tri abundance. So if you then sequence and then normalize based on read depth, most of the effects might not be clearly observable, as you see here in example A. And I'm going to show you an example from actual data. So this data comes from the very first paper that described the RX normalization technique. So in this paper, the authors treated their cells. They treated the human cells with DOT1L inhibitors. DOT1L is the enzyme that deposits K79 dimethylation. And then they mixed their cells in different proportions. So you see here, they added treated cells at 25%, 50%, 75% with untreated cells, and then performed ChIP-seq more specifically. So you can see here, even when 75% of your treated cells are added to untreated cells, the signal for K79 di is still very high and close to 80% or close to 60%, which is inaccurate. And then when a spike-in normalization is performed, you can actually now see that when 50% of cells are added, the signal is actually dropping to 50%. The signal of K79 di is proportional to the amount of treated cells. So this reflects what we call the ground truth, which you see here in this cartoon, or the true enrichment, which can be measured with another quantitative method, such as mass spec, or something that's maybe sometimes even less accurate, like a Western blot. So what's also important is that the amount of spike-in, again, whether it's chromatin to chromatin for ChIP-seq, or DNA, or cells or nuclei for immunocleavage assays, it's important that the amount of spike-in is equal across all of your samples. Because if you double up on the amount of spike-in in your treated sample or your treated cells, you're going to also overinflate the effect. as you see here in this example over here. Another thing that's very important, aside from the exogenous spiken being equal across all of your sample, is that you need to have enough, you also need to have enough spiken to be able to accurately determine the effect of your modality or the effect of your treatment. As you see here, if the amount of spiken is not equal, which often happens, it's not going to be exactly equal, but it should be as equal as possible. And then if there's a big variability in the proportion of the spiken in your samples, this is going to also skew the the data and how it looks like. So the exogenous spiken has to be easily discernible as well. That is why if you're working with mouse or human cells, it's preferred that you use Drosophila cells that can be easily distinguished from the human and mouse genomes. So this is a very nice example from this paper, and it shows you the importance of normalization and the importance of having an input, which is your non-IP'd chromatin. And to some extent, the closest equivalent then input for immunocleavage assays like CUT&RUN and CUT&TAG is called the IgG control. Of course, it's not the absolute equivalent, but it's the closest possible for this purpose. So you see here, in this experiment or in this example, cells were treated with either TSA or the MSO, and TSA acts as an HDAC inhibitor. So in the TSA condition, you'd expect the cells to have higher acetylation signal from K9AC or K27AC. However, when you do a read depth normalization, it's very hard to depict changes in acetylation. It's actually non-observable across these sample sets. And then in the second example, an input is not taken into account for spiken variation. You need to also spiken your input to make sure that you have equal amounts of spiken across your samples. Which again is basically your IgG control equivalent for CUT&RUN and CUT&TAG. So the problem here is that this case assumes that the signal will be linear with respect to epitope abundance, which is not necessarily true. You can clearly see here that the observable change is not very strong, and that the amount of spiken, as you see here on the y-axis, also affects the overall signal. And then finally, that's the close to perfect scenario when both a spiken is performed and is added to both your sample and your input. You can clearly see here that the signal is mostly conserved across the different conditions, regardless of the ratio used. And the observable differences in acetylation is also very clear between the TSA and the MSO conditions. So final recommendations for spiken. An exogenous reference has to be added, which oftentimes is chromatin for ChIP-seq, or cells or nuclei for immunocleavage assays like CUT&RUN and CUT&TAG, and it is required for accurate assessment of the epigenome. Variation in spiken to sample ratio should be minimal as much as possible, and of course, it should be accounted for using a non-IP control, which is your input, or your IgG control. And thirdly, the exogenous reference should be sufficiently quantifiable. It should be unique enough to be easily discernible when analyzing your data. So you can use Drosophila spiken. Drosophila S2 cells are commonly used when you're interrogating human or mouse cells. So the second very important consideration as well is antibody selection. You need to make sure that your antibody is specific to your target protein or your target PTM. So if possible, choose an antibody that's knockout validated to make sure it's specific. And then the second very important thing, especially for PTMs, is promiscuity. So epigenetics companies actually have very strict antibody selection criteria, but make sure to select an antibody that was tested for its promiscuity using an assay that kind of mimic a native nucleosomal context, which is similar to our experimental settings, such as the use of semi-synthetic nucleosomes, which is becoming more common and more widely used. And you can see in this example, actually, this group was testing different antibodies against K4 trimethylation. And in the set, the first two antibodies also promiscuously bind to K4ME dye at high avidity. And then when you look at these tracks, whether it's ChIP-seq or CUT&RUN, when you look at these tracks, if you look at these two antibodies that have a low specificity or medium specificity for K4-tri, they actually more resemble K4-di tracks and less that of an actual K4-tri track done with an antibody that is very specific to K4-tri. So this shows you how important antibody selection is and how it can influence or bias the results and your interpretation. Another important aspect, not just for ChIP-seq or immunocleavage assays, but for all of your assays, of course, is to prioritize the use of monoclonal antibodies when possible, as this limits the potential lot-to-lot variation, and which can otherwise complicate troubleshooting or external validation with other sample sets, with other existing data sets, or with or when your antibody runs out and you have to buy a new batch. Another important topic is the target that you're mapping. And here I'm also addressing at the same time a few concerns that we got our registration form. And thanks a lot for actually providing these questions. They were very helpful for us to tailor this presentation for your needs. So based on the target mapped, you might have to do light to moderate cross-linking, especially if your target is labile, if it's less abundant, or any other similar scenario. I would recommend that you start with 0.1% formaldehyde fixation for 1 minute or for a few minutes. You can actually do a series of for different incubation times and see how that affects your signal-to-noise ratio. I think it's also very important that you do this assay or do this optimization while also doing it on native cells at the same time. And then also, again, depending on your target mapped, for example, for labile marks like K27 acetylation or K9 acetylation, the addition of stabilizers in your buffers could help. So you can add TSA or sodium butyrate, for example. And thirdly, the addition of SDS and proteinase K significantly enhances recovery, especially when you're working with transcription factors or anything else that's lowly abundant on chromatin or on DNA. And even though most oftentimes commercial protocols recommend or suggest the use of spin filter purification, the organic extraction method results in higher yield and also higher complexity of your final DNA output. And then, of course, another thing that we were asked quite a lot is which technique to be used. CUT&RUN works for a broader range of targets, including post-translational modifications and transcription factors. So preferably if you're profiling a transcription factor or something that's lowly abundant, like a lowly abundant PTM, I would recommend that you start with CUT&RUN and see how that goes. But of course, CUT&TAG does work for some transcription factors, and that has been shown very eloquently in multiple papers. But to some extent, transcription factors tend to be less reliable for non-PTM targets. And of course, CUT&TAG has its own advantages, such as its use for single nuclei profiling, because it bypasses library prep and DNA purification. Another concern that we got, which was, of course, already part of our slides, is how does the data look like? How does it compare between the different technologies? So firstly and importantly, in situ-based cleavage technologies offer significantly higher dynamic range. You can clearly see and appreciate in this example of mapping K27 trimethylation that true biological signal from CUT&RUN and CUT&TAG is easily discernible from random experimental interference coming from a ChIP-seq. The signal here coming from ChIP-seq is sadly highly muffled by high background noise because of the caveats I spoke about before. And you can appreciate more clearly in this example as well. It's a snapshot of mapping CTCF sites. that chances of missing lower peaks, which are potentially sites of lower enrichment, is much easier when doing ChIP-seq because, again, of the low dynamic range. Moreover, if you want to pick up subtle changes, or if you have very low abundance of your mark, this can also become problematic if you perform ChIP-seq. And the same trend can also persist or can also be seen when profiling other targets such as MIC and MAX, as seen in this example. And then in this paper, if you look at the peaks that are overlapped between MIC and MAX, there's significantly higher overlap from data coming from CUT&RUN, probably because ChIP-seq fails to adequately identify MAX peaks. Lastly, an important question that we got is how does the data correlate? How does it look like? How can we compare ChIP-seq to CUT&RUN? So this is an active area of investigation, but data coming from immunocleavage assays tend to be a bit different from ChIP-seq. There's higher correlation for active marks, or at least marks that are on euchromatin, more specifically methyl marks, which could also be due to the stability of the mark itself, or because we just have better antibodies that can probe for these marks that are specific for these marks. And as I just mentioned, this study also shows that in situ cleavage assays and ChIP-seq, they do produce more or less similar profiles for marks that are demarcating euchromatin and to some extent facultative heterochromatin. But this is not really the case for canine trimethylation, for example, which is enriched in the insoluble heterochromatin fraction. And you can see further that when these loci are assigned to 3 clusters based on their enrichment pattern, clusters 1 and 2, which significantly overlap LTRs, are severely underrepresented in data generated by ChIP-seq. And this enrichment of heterochromatin or heterochromatic repetitive elements is also observed for CUT&RUN. So it's a common trend for these immunocleavage assays. And this is another inherent limitation with ChIP-seq. There's still more papers looking into how data can be correlated, data coming from ChIP-seq versus immunocleavage assays. And I think this paper and I think this paper is worth having a deeper look into. I recommend that you read this paper because it doesn't just discuss the overlap in peaks, but also puts it in a more functional perspective when comparing acetylation peaks coming from CUT&RUN, CUT&TAG, and ENCODE ChIP-seq. And I think more studies looking into correlating data coming from these different technologies are gonna keep on coming out. And then at the end, I would like to provide you with a few resources which some of you also asked for. So the detailed CUT&RUN protocol, the detailed CUT&TAG protocol, and a few optimizations such as CUT&RUN salt. If you would like to look at purifying different epigenetic complexes that might be more complicated using the standard protocol. So you can do salting out experiments from your CUT&RUN based protocol. Also ultra low input CUT&RUN that can be used for profiling embryos or profiling ultra rare cell populations, in addition to more optimizations that can be performed depending on the assay of interest or the experiment of interest. And at the end, I would like to acknowledge my labs, of course, acknowledge my funding partners that have been great support for my doctoral research. Of course, CEEHRC and the CEEHRC trainee committee that provided a lot of important input and helped develop this presentation. And of course, I would like to thank you for your attendance and for providing us with questions that help prepare these slides tailored to your needs. And I think because we're not doing too bad on time, maybe I can address a few more questions that I got from the registration form. I added those today morning. So those are some of the newest questions we got, and many of them are or were repetitive. So the first question was about whether qPCR analysis can be used on CUT&RUN samples to make sure that the assay worked. Yes, of course, CUT&RUN or CUT&TAG success can be analyzed using qPCR or droplet digital PCR. However, unlike ChIP-seq, because the fragments here, because it's an in situ cleavage, the fragments here are very small. So it requires that you do your library prep before. So you need to prepare your library for CUT&RUN more specifically before you can actually do a qPCR or a ddPCR to amplify short fragments of DNA. And even though some commercial protocols recommend that you quantify your output, so quantify the amount of DNA you get at the end before library prep, it might not always work, especially if you're doing some sort of cross-linking, some form of cross-linking, or if your target is not highly abundant. So I would still say, even if your enriched DNA has similar or sometimes even less amounts than your IgG, I would say still do a qPCR-based analysis or a ddPCR-based analysis, or even send for sequencing, because it's oftentimes with lowly available or lowly abundant targets that it's going to be very hard to quantify the output DNA with fluorometric approaches. And then another question was if it's possible to profile multiple transcription factors or histone modifications at the same time. Of course, this is done. This can be performed with barcoded antibodies, and I've attached a QR code for a paper if someone's interested in looking at the protocol. And also another point here, a brief reminder that this video will be available on YouTube, so you can have your QR codes for you to visit later on. Another question was the best technique to measure viability. As I said, trypan blue exclusion assay is more than enough for both cells and nuclei. Some people prefer using more fluorescent-based methods for nuclei. They both work almost equally well. Just make sure that your cells are healthy and that they're not clumped. Same goes for nuclei, which often is an issue. And then, of course, CUT&RUN or CUT&TAG can be performed for non-model organisms. This has been done. There are papers about it. There's even protocols modified for plant cells. So it's doable. You just need an antibody that can bind your target with high avidity, that's not very promiscuous, and that you have a genome to act as a reference for mapping. And then again, for short-lived transcription factor DNA interactions, that's where you have to do some form of cross-linking. Some labs also prefer doing this on nuclei for TF interactions. With cross-linking, from my experience at least, we had better successes. And that's about it. Thanks again. And in case anyone has more questions, I'm happy to answer. I'll have a look at the Q&A chat box in a bit.
Thanks, Wajih. That was an excellent presentation. I just have a couple announcements before we get into more questions from the Q&A box. So a reminder to everyone that this is session 1 of 2, so please register for the next session. I put the link in the Q&A. And CEEHRC is also looking for everyone's feedback to help with future funding applications to support the sustainability of the network. So we have a survey both in English and French, and that's also in the Q&A.
Thank you, Signy. So I'm looking at the questions. Many of it was addressed, thankfully. I had a few questions for CUT&RUN and CUT&TAG on tissues. I think for tissues, if you're able to dissociate your cells with standard protocols, you can still do it on cells. That's something that we've done for mesenchymal tumors using standard collagenase and dispase-based approaches. So if that works well for you, if you get good viability, then that's okay. If not, I would say snap freeze your tissues and then isolate nuclei. You don't want to have any apoptotic cells or lysed nuclei in your samples, as they will keep on releasing genomic DNA during the process. So that's going to contribute to a lot of noise. If you want to have crisp reads, you need to make sure that the cells and nuclei are healthy and that they're not lysed. So yes, for tissue CUT&RUN and CUT&TAG, especially if you have a big experiment, you're collecting tissues from patients, you're collecting tissues from mouse models, you have plenty to work with. I would say don't incubate your tissues because in most cases they're not protected, especially for labile marks. You're going to lose a lot of acetylation marks if you want to probe for acetylation. So just snap freeze your tissues and then isolate nuclei with standard protocols. So there's a lot of protocols available online, and I think some of the protocols I had attached in the QR codes also includes different methods to isolate nuclei, which is more or less standardized because it's also used for ChIP-seq and for other purposes. And as I said, a form of modification of a protocol for transcription factors is to perform light cross-linking and see how that goes. It worked really well for us, even with just 0.1% for 1 minute. It made a huge difference in enhancing the signal-to-noise ratio. Digitonin is used for both cells and nuclei. As I said, the first step is going to be performed under native conditions. Someone was asking if digitonin is going to be used only for nuclei. No, digitonin is used for both cells and nuclei. Yeah, in both cases. But the first step is done under native conditions for CUT&RUN in most protocols, and then digitonin is added in the antibody buffer and in subsequent washes. Mouse primary antibody does work as well as rabbit in many cases. And the protocol, thanks to the Hennikov lab, the protocol has been optimized to work for both because now the MNase is fused to both protein A and protein G. So it works well for both. Just check with the manufacturer recommendations and make sure that, again, it's specific to your target and it does not exhibit a lot of promiscuity. And yes, as someone was asking, the recording will be available. It's going to be uploaded on YouTube, so you're going to have access to the different snapshots and different QR codes if you're interested in looking at protocols and looking at other publications that use these technologies. I think that was everything.
Well, thanks, Wajih. That was really, really excellent. If we don't have any further questions, I think we can wrap up this seminar. And just remember, please attend the next seminar next week on the bioinformatics side of CUT&RUN and CUT&TAG techniques. Thanks, everyone.
Thank you.